Techniques in Cultivating and Identifying Animal Viruses, Chapter 5, Topic 5.4. In this topic, we're going to be discussing the difference between in vivo and in vitro cultivation of viruses. We're going to be talking about live animal cultivation, bird embryo cultivation, and cell culture techniques. At the end of this recorded lecture, you should be able to list the three principal purposes of cultivating viruses, describe three ways in which viruses are cultivated. Viruses, as intracellular parasites, must be cultivated in living cells. For a long time, animal viruses could only be grown in cells that were in the intact animal. This is called growing them in vivo. Techniques were developed for growing animal cells in the lab in flasks in a single sheet, or monolayer. When viruses are added to these cell cultures, the viruses are said to be cultivated in vitro. We go to all this effort for three reasons: to isolate and identify viruses in clinical specimens. I did that for quite a few years. Prepare viruses for vaccines, and to do detailed research on viral structure, multiplication cycles, genetics, and effects on host cells. Viruses can be grown in any lab animal, be they cute fuzzy mammal or creepy crawly bugs. Because viruses are very specific in what host cells they will infect, you have to use the proper animal for the proper virus. For example, there's no point in infecting a cat with a virus that will only infect swallowtail butterflies. You have to use swallowtail butterflies if you're going to study a virus that infects them. Some viruses can be grown in fertilized bird eggs, or, more specifically, in bird embryos in the fertilized bird eggs. They are self-contained and don't need to be fed or their cages cleaned. They just need to be kept at the correct temperature and humidity and turned on a regular basis. Chicken embryos are used to grow large numbers of influenza viruses for the yearly vaccines. It would be impractical to try to do the same thing with chicks or adult chickens, if only from a space and potential contamination perspective. Once we developed the ability to grow animal cells outside of the animal, cultivating viruses has been much easier. Tissue cultures require much less space and care than even bird embryos. You do have to change their media, keep them at the proper temperature and O2-CO2 ratio, and prevent them from becoming contaminated with either bacteria or fungi. They don't have an immune system after all. Today, a lot of virus research is conducted using cell culture techniques. That's it for this topic. You should now be able to list the three principal purposes of cultivating viruses and describe three ways in which viruses are cultivated.